So for the next topic for water pollution, it's all about water pollution in rivers. So there will be 9 subtopics, and these are total maximum daily load, effect of oxygen demanding waste in rivers, biochemical oxygen demand, graphical determination of BOD contents, laboratory measurement of BOD, additional notes on BOD, nitrogen oxidation, Streeter-Phelps DO subcurve, and effects of nutrients on water quality in rivers. So for the introduction, the objective of water quality management is simple to state: to control the discharge of pollutants so that water quality is not degraded to an acceptable extent below the natural background level. However, controlling waste discharges must be quantitative endeavor. So, what does this quantitative endeavor imply? It means we must be able to measure the pollutants, predict the impact of the pollution on water quality, determine the background water quality that would be present without human intervention, decide the levels acceptable for intended uses of the water. So the impact of pollution on a river depends both on the nature of the pollutant and the unique characteristics of the individual river. What are these characteristics? These characteristics includes the volume and speed of water flowing in the air, in the river's depth, the type of bottom, the surrounding vegetation. So here are some of the factors that water quality management considers about water pollution in rivers: climate of the region, the mineral heritage of the watershed, land use patterns, and types of aquatic life in the river. So for the first subtopic, it's all about total maximum daily load, or TMDL. A TMDL specifies that maximum amount of pollutant that a water body can receive and still meet water quality standards. In addition, the TMDL allocates pollutant loadings that may be distributed among point and non-point sources. The TMDL computation is defined as TMDL is equals to summation of waste load allocations plus summation of load allocations plus margin of safety. So software that helps in developing TMDL. A system software called Better Assessment Science integrating point and non-point sources or basins is used to develop TMDL. Basins integrates a Geographic Information System, or GIS, national watershed and meteorological data, and state-of-the-art environmental assessment and modeling tools. So that's for TMDL. So for the next subtopic, it is all about the effect of oxygen demanding waste on rivers. The introduction of oxygen demanding material, either organic or inorganic, into a river causes a depletion of the dissolved oxygen in the water. This poses a threat to fish and other higher forms of aquatic life if the concentration of oxygen falls below a critical point. All about predicting oxygen depletion. To predict the extent of oxygen depletion, it is necessary to know how much waste is being discharged and how much oxygen will be required to degrade the waste. However, because oxygen is continuously being replenished from the atmosphere and from photosynthesis by algae and aquatic plants, as well as being consumed by organisms, the concentration of oxygen in the river is determined by the relative rates of these competing processes. About measuring organic oxygen demanding material. Oxygen Organic oxygen demanding materials are commonly measured by determining the amount of oxygen consumed during degradation in a manner approximating degradation in natural waters. So that's all for the second subtopic. So for the third subtopic is all about biochemical oxygen demand. The theoretical oxygen demand, THOD, or amount of oxygen required to oxidize a substance to carbon dioxide and water, may be calculated by stoichiometry if the chemical composition of the substance is known. For computing THOD, we begin by writing a balanced equation for the reaction, and next is to compute the gram molecular weight of the constants. Next is chemical oxygen demand. In contrast to THOD, the chemical oxygen demand is a measured quantity that does not depend on knowledge of the chemical composition of the substances in the water. In the COD test, a strong chemical oxidizing agent is mixed with the water sample and then boiled. The difference between the amount of oxidizing agent at the beginning of the test and that remaining at the end of the test is used to calculate the COD. Next is all about biochemical oxygen demand. If the oxidation of an organic compound is carried out by microorganisms using the organic matter as a food source, the oxygen consumed is known as biochemical oxygen demand. The actual BOD is less than the THOD due to the incorporation of some of the carbon into new bacterial cells. The BOD test is an indirect measurement of organic matter because we actually measure only the change in dissolved oxygen concentration caused by the microorganisms as they degrade organic matter. THOD, COD, and BOD. Only under rare circumstances will the THOD, COD, and BOD will be equal. The chemical composition of all the substances in the water is known and they are capable of being completely oxidized both chemical and biologically. Then the three measurements of oxygen demand will be the same. So for the BOD rate equation, BOD is equals to oxygen equivalent of organic compounds at time t equals 0 times 1 minus 10 raised to negative kT. For the numerical value of the rate constant is dependent on the following: the nature of the waste, the ability of the organisms in the system to utilize the waste, and the temperature. So that's all for the BOD. Now for the fourth subtopic is all about graphical determination of BOD constants. A variety of methods may be used to determine K and LO from an experimental set of BOD data. The simplest and least accurate method is to plot BOD versus time, and the rate equation is used to solve K. One simple method around this is called Thomas graphical method. So for the first step, with the experimental results of BOD for various values of T, calculate T over BOD dt raised to 1/3. And next, plot the computed value versus T on a arithmetic graph paper and draw the line of best fit by eye. Third, determine the intercept A and slope B from the plot. For determining intercept A is equals to quantity K LO raised to negative 1/3. For slope B is equals to quantity K raised to 2/3 over 6 times quantity LO raised to 1/3. For the fourth step, calculate K and LO. To calculate K is equals to 6 times quantity B over A. And to calculate LO is equals to 1 over 6 times quantity A squared times quantity B. So that's all for the fourth subtopic. So for the fifth subtopic, it is all about laboratory measurement of biochemical oxygen demand. In order to have as much consistency as possible, it is important to standardize testing procedures in measuring BOD. So I will explain to you each steps for laboratory measurement of biochemical oxygen demand. So based on the sample given, the bottle will be stoppered to exclude air bubbles. Samples require dilution because the only oxygen available to the organisms is dissolved in the water. The most oxygen that can dissolve is about 9 milligrams per liter. So the BOD of the diluted sample should be between 2 and 6 milligrams per liter. Samples are diluted with special dilution water that contains all of the trace elements required for bacterial metabolism, so that degradation of the organic matter is not limited by lack of bacterial growth. The dilution water was also contains an inoculum of microorganisms so that all samples tested on a given they contain approximately the same type and number of microorganisms. So for the figure shown, the bottle on the left is shown with the cap removed to illustrate the shape of the bottle glass stoppers. The point on the end of the stopper is to ensure that no air is trapped in the bottle. The bottle in the center is shown with the stopper in place. Water is placed in the small cup formed by the lid. This acts as seal to further exclude air. The bottle on the right is shown with plastic wrap over the stopper. This is to prevent evaporation of the water seal. So for the ratio of undiluted to diluted sample is called the sample size, usually expressed as a percentage, while in the inverse relationship is called the dilution sample factor. So mathematically, these expressed are the appropriate sample size to use that can be determined by dividing 4 milligrams per liter by the estimated BOD concentration in the sample being tested. A convenient volume of the undiluted sample is then chosen to approximate to this sample size. Now, next is step 2. Blank samples containing only the inoculated dilution water are also placed in BOD bottles and stoppers. Blanks are required to estimate the amount of oxygen consumed by the added inoculum of microorganisms called seed in the absence of the sample. Step 3. Stoppered BOD bottles containing diluted sample and blanks are incubated in the dark at 20 degrees Celsius for the desired number of days. For most purposes, a standard of time of 5 days is used. To determine the ultimate BOD and the BOD rate constant, additional times are used. The samples are incubated in the dark to prevent photosynthesis from adding oxygen to the water and invalidating oxygen consumption results. As mentioned earlier, the BOD test is conducted at a standard. Next is step 4. After the desired number of days has elapsed, the samples and blanks are removed from the incubators and the dissolved oxygen concentration in each bottle is measured. The BOD of the undiluted sample is then calculated using the following equation. BODT is equals to dissolved oxygen concentration in blank after T days of incubation minus dissolved oxygen concentration in sample after T days of incubation times dilution factor. So that's all for the 5th subtopic. So for the 6th subtopic, it is all about additional notes on biochemical oxygen demand. Although the 5-day BOD has been chosen as the standard value for most wastewater analysis and for regulatory purposes, ultimate BOD is actually a better indicator of total waste strength. For any one type of waste having a defined BOD rate constant, the ratio between ultimate BOD and BOD5 is constant so that BOD5 indicates relative waste strength. For different types of waste having the same BOD5, the ultimate BOD is the same only if, by chance, the BOD rate constants are the same. The 5-day BOD was chosen as the standard value for most purposes because the test was devised by environmental engineers in England, where rivers have travel times to the sea of less than 5 days, so there was no need to consider oxygen demand at longer times. Since there is no other time that is any more rational than 5 days, this value has become firmly entrenched. That's all for the 6th subtopic. So for the 7th subtopic, it's all about nitrogen oxidation. Many organic compounds, such as proteins, also contain nitrogen that can be oxidized with the consumption of molecular oxygen. Two processes must be considered separately because the mechanism and rates of nitrogen oxidation are distinctly different from those of carbon oxidation. Logically, oxygen consumption due to oxidation of carbon is called carbonaceous BOD, while that due to nitrogen oxidation is called nitrogenous BOD. The nitrogen is released into the surrounding water as ammonia. At normal pH values, this ammonia is actually in the form of the ammonium cation. The ammonia released from organic compounds, plus from the other sources such as industrial waste and agricultural runoff, is oxidized to nitrate by a special group of nitrifying bacteria as the source of energy in a process called nitrification. The rate at which the NBOD is exerted depends heavily on the number of nitrifying organisms present. In untreated sewage, there are few of these organisms, while in a well-treated effluent, the concentration is high. When samples of untreated and treated sewage are subjected to the BOD test, oxygen consumption is going to increase. In the case of untreated sewage, the NBOD is exerted after much the CBOD has been exerted. In the case of the treated sewage, a higher population of nitrifying organisms in the sample reduces the lag time. When measurement of only carbonaceous BOD is desired, chemical inhibitors are added to stop the nitrification process. The rate constant for nitrification is also affected by temperature. So that's all for the sixth subtopic. For the next subtopic, it is all about Streeter-Phelps DO subcurve. One of the major tools of water quality management in rivers is the ability to assess the capability of a stream to absorb a waste load. This is done by determining the profile of DO concentration downstream from a waste discharge. This profile is called the DO subcurve because the DO concentrations dips as oxygen-demanding materials are oxidized and then rises again further downstream as the oxygen is replenished from the atmosphere. To develop mathematical expression for the DO subcurve, the sources of oxygen and the factors affecting oxygen depletion must be identified and quantified. For the sources of oxygen, the only significant sources of oxygen are from aeration from the atmosphere and photosynthesis of aquatic plants. Oxygen depletion is caused by a larger range of factors, the most important being the BOD, both carbonaceous and nitrogenous, of the waste discharge, and the BOD already in the river upstream of the waste discharge. For factors affecting oxygen depletion, the second most important factor is that the DO in the waste discharge is usually less than that in the river. Thus, the DO at the river is lowered as soon as the waste is added, even before any BOD is exerted. Other factors affecting dissolved oxygen depletion include non-point source pollution, the respiration of organisms living in the sediments, and the respiration of aquatic plants. So, there will be 13 subtopics for the qualitative discussing about the other factors affecting the DO levels, and these are. And now for the last subtopic for water pollution in rivers, and this is all about effects of nutrients on water quality in rivers. Although oxygen-demanding wastes are the most important river pollutants on an overall basis, nutrients can also contribute to deteriorating water quality in rivers by causing excessive plant growth. Some plant growth is desirable, since plants form the base of the food chain and thus support the animal community. However, excessive plant growth can create a number of undesirable conditions such as thick slime layers on rocks and dense growths of aquatic weeds. So there are two effects. First is the effects of nitrogen. There are three reasons why nitrogen is detrimental to a receiving body. First, in high concentrations, NH3N is toxic to fish. Second, NH3 in low concentrations and NO3 serves as nutrients for excessive growth of algae. The conversion of NH4 to NO2 consumes a large quantities of dissolved oxygen. Second, effects of phosphorus. The major deleterious effect of phosphorus is that it serves as a vital nutrient for the growth of algae. In the phosphorus availability meets the growth demands of the algae, there is an excessive production of algae. When the algae die, they become an oxygen-demanding organic materials as bacteria seek to degrade them. This oxygen demand frequently overtaxes the DO supply of the water body and, as a consequence, causes fish to die. So that's all for the ninth subtopic and for the water pollution in rivers.